When we talk about the immune response by the body, what stimulates the adaptive immune system into action are antigens. And antigens are non-self particles or foreign cells that are identified by the body. So they have to be identified as non-self. Recall that we talked about antigens with the blood types, and the antigens on the red blood cells that we are born with are not considered non-self or foreign, so our immune system will leave them alone. So any substance that mobilizes the adaptive defenses and invokes an immune response are antigens. So most of our antigens are large, complex molecules that are normally not present in the body. So sometimes, though, we have smaller particles that are also antigens, but rather than us attacking them to prevent infection, the immune system is mobilized and causes an allergic reaction. So in the case of people that are allergic to penicillin, that should not be considered a harmful substance. It's there to benefit us, to kill bacterial infections or treat bacterial infections, but as a result, our immune system responds to it, and we end up with hypersensitivity reactions like rashes, narrowing airways, and just allergic reaction or hypersensitivity. So those antigens, examples again are penicillin, poison ivy, animal dander. Some people are allergic to certain cosmetics or detergents in their laundry. So we really have to be careful sometimes of things that are normally not a problem actually serving as an antigen and stimulating the immune response. So if you look at an antigen, multiple antibodies can bind to the antigen depending on those binding sites. So we call these determinants. It's just the shape of the chemical shape of the antigen. It has specific binding sites for specific antibodies. But now these antibodies, it takes time to develop those once they encounter an antigen. And we'll talk a little bit about the primary and secondary immune response in how we form antibodies in a few slides coming up. So we have special cells called antigen-presenting cells. And these cells are specific cells that present antigen for either killing or stimulating other cells to create antibodies against it. But either way, they present antigen for stimulation and attack by the immune system. So there's three types of antigen-presenting cells. They're called dendritic cells, macrophages, and B cells. They all act as antigen-presenting cells. So they will take the antigen, bring it in by phagocytosis, and then just present fragments of those antigens on the surface where they can be recognized again by other cells of the immune system. And for example, T cells are cells that will recognize these antigens on the surface of the antigen-presenting cells. So here's an example of a dendritic cell with these special cellular processes and presenting antigen on the surface. So moving into the next branch of the adaptive immune system, or the first branch of the adaptive immune system, is humoral immunity, and the major cells involved in humoral immunity are the B cells. So it's important to understand that this is the branch of the immune system in which we make antibodies for specific antigens that we encounter. So when you hear the word humoral immunity, I want you to think antibodies are formed. So specific things we can say about humoral immunity is, again, you have antibodies produced by the B cells, and those antibodies then are freely floating in circulation, in the plasma. We talked about the antibodies that we form against foreign blood antigens, like a person that is A blood type will have B antibodies in the plasma. So we find these antibodies in the plasma, and they're formed when we encounter a foreign antigen. And those antibodies will bind to the target cell and destroy it. So again, B lymphocytes are a type of lymphocyte that produces antibodies, and they help to activate themselves when they present antigen to a T cell. So we talked about T cells recognize antigen-presenting cells, and that's what happens with a B cell, is it encounters antigen and presents it to a T cell, and then that T cell will activate it. So when we look at T cells and B cells, the way they're named depends on how they or where they develop. So we know that in the red bone marrow is where our white blood cells are produced. When they enter the bloodstream then, after they are produced, the T cells mature in the thymus and become able to recognize self from non-self, and the B cells mature and recognize self from non-self in the bone marrow. So that's where they get their name. T cell for maturing in the thymus, which starts with a T, and B cells mature in the bone marrow, which starts with a B. So B cells, T cells is where they get their name. So they mature there. And then after they mature, they enter the bloodstream and are infiltrate the lymph nodes and the lymph organs. And they remain there where they will encounter antigens that might be flowing through the lymph. So we talked a little bit about the lymphatic system, or there's a PowerPoint that I posted on the lymphatic system that you should have reviewed. If you haven't, just know that lymph is the fluid from the interstitial fluid that enters the lymph vessels, and then the lymph nodes filter that lymph as it passes through the lymph vessels and then returns that filtered plasma back to the general circulation. So when we have these T cells, B cells present in the lymph node, they can become activated when they bind antigen, specifically the B cells, and then they multiply in number. So B cells, when they encounter antigen in a lymph node, will multiply in number and form memory cells. And effector T cells are also formed, and then these will enter the blood and be throughout the body waiting to attack when the antigen is encountered again. So when we have initial exposure to an antigen, so let's say when you first get chicken pox, for example, the chicken pox, well, let's look at yeah, that's a good example. We'll just go with the chicken pox. So we encounter the chickenpox virus. It enters our cells. We form antibodies to that chickenpox virus, and then that will activate the B cells, and some of the B cells will form plasma cells, which are effector B cells, to secrete a lot of antibodies, and those antibodies will then bind to other virally infected cells and destroy them. So this antibody production is important to taking care of the illness when we actively have the chickenpox. So this usually takes a week or two to completely destroy the virus and form antibodies for the next response. So that's the primary response when we first encounter an antigen. So it takes about 3 to 6 days for us to produce enough antibody to really attack those antigens. So that's why we're sick for a while when we first encounter an illness. And then at about 10 days, we have a peak level of antibodies, and then those levels decline again. But if we are exposed to that same antigen, we have memory cells that were produced during the primary response. Those memory cells then respond very quickly within hours of exposure, and we have a lot of antibodies within 2 to 3 days, so we don't get sick because of that. So the second exposure to chickenpox virus, for example, you're not going to get sick because you have already produced the antibodies the first time you were exposed, and those memory cells are ready and waiting to produce more antibodies upon the second exposure. So those antibody levels can remain high for quite a while, and that's why sometimes when we give immunizations, we require boosters to make sure we get that secondary response where we have good antibody production. And even then, we sometimes need boosters 10 years later, like we do that for tetanus, for example. We want to make sure we have good antibody response, so we want to boost tetanus every 10 years. So here's a little graph showing the difference between the first exposure to an antigen compared to the second exposure. You can see producing a lot of antibodies in the beginning, but it kind of tapers off after about 10 days and then goes back down And then a month later, if you're exposed again, we get this huge amount of antibody production, and you're not going to get sick because of that, because those antibodies are right there to attack those virally infected cells and destroy them. So when we talk about humoral immunity, we're talking about antibody production. So there's different ways that we can form antibodies or treat exposure to antigen. And the active form means, and it's important that you know the difference between active and passive. Active means that the body is actively making the antibodies themselves. So the difference between active and passive humoral immunity: active, the person is making the antibodies, and in passive, the person is receiving antibodies from someone else who made the antibodies. So you're receiving antibodies that you did not make in the passive branch, and when you're actively making the antibodies yourself, that's the active branch. So naturally acquired active humoral immunity is when you get sick. You're exposed to chickenpox virus, you make the antibodies against it because you acquired that antigen and made those antibodies because you're exposed in nature. So when you go to school, there's a kid with chickenpox, you get the chickenpox, that's naturally acquired active immunity. If, however, you didn't encounter the virus naturally, but you countered it artificially, as in an injection, like when you get the flu shot or your MMR vaccine or a tetanus shot, that is artificially acquired active immunity. You're still going to make antibodies, but you weren't infected with the illness. That wasn't how you were exposed. You were artificially injected with an antigen or parts of the antigen to stimulate antibodies. So it's still active because you made your own antibodies, but the way you were exposed to the antigen was artificial through injection. So the passive branch, we said, is when you're not actively making the antibodies, but you're using those antibodies to fight infection. So a baby that is nursing from its mother, the mother made the antibodies, those pass into the milk, and the baby acquires that immunity to those illnesses through the breast milk. So this is really a benefit of breastfeeding, is babies in those early months of life are not going to get those illnesses because they will be immune to all the illnesses that the mother is immune to because of having her antibodies being passed through the breast milk. Artificially required passive immunity is when you're not getting them naturally through breastfeeding, but you're getting them artificially through injection. For example, if you get bit by a rattlesnake and you need the anti-serum, which anti-serum means antibodies. So you would receive an injection of those antibodies to actively fight the infection. Because we don't want to waste time waiting for you to produce the antibodies because you've been, bit by a rabid dog or bitten by a rattlesnake. We want to make sure that we give you those antibodies right away to fight the infection so you don't become ill or die. So no examples of each of the different types of humoral immunity, active versus passive and natural versus artificial. So there'll be several different questions of that on the exam. So again, antibodies, we find these in the plasma of our blood. They are specific proteins, and they're made by special cells called plasma cells, which are types of B cells that have changed to plasma cells after they've been exposed to an antigen. So they bind to antigen, and the B cells, again, help specifically, I should say, develop antibodies that recognize specific antigens. And there's 5 different classes of antibodies. So we talked about primary response already. In the secondary response, you can see the number of antibodies produced here when we're first exposed compared to the number of antibodies when we're exposed a second time, much larger, and that's why we don't become sick. But let's look at the 5 classes of antibodies here. We can see that what's unique about each one, the IgM, this is the first antibody to be secreted during the primary response. We can see it's a very large antibody, has lots of binding sites. So this is an important antibody that we see in the plasma of our blood. The next one, the IgA antibody. This is an antibody that we will find in some of our body secretions, such as saliva, sweat, your intestinal juice, and breast milk. So it's the IgA specifically that we find in those secretions. And then IgD is a specific antibody that we find on the surface of B cells, and they serve as a receptor for B cells to bind antigen. Oops. And the IgG is our most abundant antibody. So most of the antibodies we find in the plasma are IgG antibodies, and they come into play after the primary response. So we said IgM was mostly effective in the primary response. The IgG comes in later at the end of the primary response and in the secondary response. So these are good at binding bacteria, viruses, toxins that are circulating in the blood and lymph. So this is what also allows the mother to pass on these antibodies to the baby through the breast milk, or sorry, through the placenta during fetal development. So these cross the placenta and protect baby when baby's first born. So those are very, very important IgG antibodies. And then the IgE antibodies, these can be the troublemakers, because if someone has allergies, they have increased numbers of IgE antibodies that bind to pollen and dog dander and those things that normally shouldn't cause trouble. The IgE antibodies will be sensitized to those. So we see a lot of inflammation and histamine release, which causes swelling of the airways, runny nose, itchy, watery eyes, rashes, because of the IgE. So when you think of IgE, think of allergies. So here's an example of how we counter antigen and antibodies once they've been formed. What happens is the antibody binds the antigen, and a couple of different things can happen involving antibodies. They can neutralize and destroy the antigen. Here we see red blood cells are being bound by IgM and causing clumping, which is bad, agglutination. Sometimes if we have free antigens, it can just pull it out of circulation and cause it to precipitate or become soluble. Rather than being insoluble and dissolved in the blood, they actually form these complexes which are visible in the blood. Or they activate a special protein called complement, which fights inflammation, and when they bind, when antibodies bind, it helps to get complement activated. And all of these complement is important for killing bacterial cells, promoting inflammation, and promoting phagocytosis by some of those macrophages that we talked about that are present in the tissues, like in the liver and the lungs. So all of this antibody antigen binding is going to promote inflammation, histamine, phagocytosis, cell destruction. And in the case of cells, that are bound or infected with foreign antigen, those are all good things that help protect our body from infection. But in the case of allergies and hypersensitivities or autoimmune diseases, these processes can really disrupt homeostasis, and then we need to look at medications to block the immune response. So again, antigen-antibody complexes don't necessarily destroy the antigens themselves, they just prepare them for destruction, which means if you look here, the antibodies bind the antigen, and then it enhances these three different processes that can occur. So the antibodies themselves don't do anything, but they just bind to the antigen, and from there the different processes of the immune system take over. So we don't see antibodies, we see them in the plasma, so the infection takes place in the plasma unless there's a specific lesion that connects the plasma to the tissues, so like an open sore. And they can act inside cells if they're attached to the virus before they enter. So that's where we can sometimes see cell destruction of antibodies if that antigen was taken with an antibody, taken into the cell with an antibody attached to it. So the last branch, the second branch of the adaptive defenses talks about cellular immunity. And when we're talking about cellular immunity, we're talking about T cells. And T cells cannot see free antigens or antigens that are floating around in the plasma. T cells can only mount an immune response if an antigen is presented by an antigen-presenting cell. And that's really important to understand, is the humoral immunity can recognize free antigens. Cellular immunity cannot. Cellular immunity means that we have cell-to-cell communication, but we can't recognize free antigens. So we really need both branches to recognize the antigen and present it to the T cells to get that final and complete immune response. So again, T cells are good for antigens that are found inside cells, because then the T cell can, the antigen presenting cell can present that antigen, and then the T cell can recognize it and release chemicals and mount a full immune response. So some T cells will directly kill the cell, those are cytotoxic T cells, and other cells will release chemicals called interleukins that will help to destroy and mount the immune response. So T cells can directly kill a cell, and they can release chemicals, like we said, to promote the immune response. And like I said, interleukins and cytokines are things that can actively kill a cell that's been invaded by an antigen. So here we see, looking at kind of some of the processes here. Here we have dendritic cell or B cell is ingesting an antigen, and then it's presenting that antigen on the surface. So remember, it breaks it down into small fragments and then presents a fragment on the cell surface, and the T cell binds to that and releases cytokines once it binds to that antigen on the surface of the B cell in this case, or any antigen presenting cell. So it releases the cytokines, and then those cytokines will stimulate other T cells and will also help to invade other infected cells. And then some of those T cells are stimulated then to become killer cells, which we call cytotoxic T cells, which will look for other cells infected with virus. And some will become helper cells, which will secrete cytokines and attract other white blood cells to the area to help fight the infection. So these T cells are really important in that they have to interact with an antigen-presenting cell that is presenting antigen on the surface, and that activates those T cells and helps them to either become helper T cells or cytotoxic T cells. So again, we have helper T cells and the cytotoxic T cells, which we'll talk about here, but there's two types, CD4 T cells and CD8 T cells, and these are the ones that can directly attack. So that's why they're called cytotoxic T cells. And these are the helper T cells, which help the other cells in the immune system come into the scene and mount an immune attack. So T helper cells don't directly kill, but they stimulate the immune response, where the cytotoxic T cells can destroy cells with foreign antigen inside them. So again, we have an immature T cell here. In the thymus, they become either CD4 or CD8, so these are the helper T cells, these are the cytotoxic cells, and they are activated by antigen-presenting cells presenting antigen on its surface, and we have these helper T cells being produced, or cytotoxic T cells produced, and we find those in the plasma. So the T cells, we said, mature in the thymus. They are stimulated by antigen that they encounter in the lymph node, so this is where this process occurs, and then they go down each of their cell lines depending if they are CD4 or CD8 T cells. So this just shows, again, we have an antigen-presenting cell, presenting antigen, the T cell binds to it, releases the different proteins, which in this case might be interleukins, which it says here, and that stimulates this B cell to produce antibodies. So the T helper cell helps the B cell become activated to make antibodies. So here we can see antibodies are being produced on the surface of this B cell. So the cytotoxic T cells, which we call CD8 cells, require T helper cells to activate them into the destructive kind of cytotoxic T cell. So here we see a T cell, CD8, and here's a helper T cell. Together, they bind to this antigen-presenting cell, and the interleukins produced by the helper T cell will activate this to become a killer T cell, where it can actively go and recognize and kill virally infected cells. So important that we see the helper T cell activates B cells, and it activates these cytotoxic T cells. So when you look at cytotoxic T cells, we're looking at virally infected cells, intracellular antigens. So intracellular antigens are destroyed by cytotoxic T cells. Cancer cells are considered foreign cells, and because the DNA is changed, and cytotoxic T cells can attack cancer cells. Foreign to the body, but like our food, we have a lot of foreign antigens we bring into the body, but specific foods like peanuts and seafood seem to be common antigens that are considered foreign and will stimulate an immune attack by the body. So pollen from different trees, particularly ragweed plant, is problematic for people that have fall allergies. And pet dander are examples of hypersensitivity reactions. So there's different types of hypersensitivity reactions. The type 1 is immediate hypersensitivity, which means the first exposure is no problem. We're exposed to it. We don't have any reaction, but we do form many IgE antibodies against that antigen. Then on the second exposure, say example a bee sting, someone gets stung again by a bee after they've formed these antibodies, then those antibodies on the surface of mast cells and basophils will cause a huge immune response, which will cause swelling of the airways, low blood pressure, vasodilation causes the low blood pressure, and you end up with anaphylactic shock. So very important that people that have this type of sensitivity, that they carry an epinephrine pen with them, an EpiPen. So we give them epinephrine, and that will help to dilate the airways and prevent them from losing the ability to breathe. So another type of hypersensitivity are the type 2 and 3 sensitivities. These are subacute, takes a little longer to develop, but we see antibodies binding directly to body cells in the case of the type 2. So an agglutination reaction we see if we had mismatched blood types would be an example of a type 2 reaction. A type 3 reaction would be inflammation primarily that is caused by antibodies binding to foreign antigen in a body tissue, for example in the lungs, and that causes an immune response causing lots of inflammation and trouble breathing. So farmer's lung, which is caused by exposure to moldy hay, is an example of a type 3 hypersensitivity. A type 4 hypersensitivity is caused by T cells, not by antibodies, but specifically T cells mount the response. And what happens is that we have a little longer time for the symptoms to show up, 1 to 3 days, because it's not antibody mediated. The T cells take time to get the inflammation and tissue damage going. So the inflammation again is caused by cytokines that are activating macrophages in the tissues and cytotoxic T cells. And examples of this would be those skin reactions that we see that sometimes take a couple days to show up. For example, poison ivy, when people change cosmetics or change chemicals in their detergents or deodorants, they might develop skin rashes. So it takes a while for that to show up. And the TB skin test is where we put a little bit of that antigen underneath the skin, and we usually have people come back within a set number of days to look and see if a reaction occurred. Because again, it takes a while for those T cells to mount an immune response. So we call those delayed hypersensitivities. So as we age, the immune system, what we find is that it's not as effective as what it once was able to do. So we see people as they age more subject to pneumonias, and therefore we recommend the elderly get their pneumococcal vaccine. Very important over the age of 65 that everybody have the pneumococcal vaccine. And also the flu shot, very important for people as we age, because the immune system is just not as good at fighting off those viruses, and even a common cold can turn into pneumonia because of waning immune response. We also see greater incidences of cancer, again, because of those cytotoxic T cells not being as effective on identifying and killing cancer cancer cells. As a result, we see higher incidence of cancers, in each body system. So we don't exactly know why that happens as we age, but we know that in general, all body systems tend to slow down. But we do know that the thymus, which is where T cells mature, we do know that the size of the thymus decreases significantly after childhood, and it turns to fat. So you don't even see the thymus gland on the surface of the heart visible in adults because it atrophies, but we do see a larger thymus gland on the top of the heart in children. So that might be why, because of the decreased maturing of those T cells. So that concludes our discussion of the immune system. I encourage you to look through, read chapter 21 if there's any areas you want more clarification on. And then you can take the take-home Blackboard exam on the immune system and lymphatic system.